Hello, everyone. Welcome to our lecture on tissues. This is a big one, right? There are a lot of slides, a lot of material in tissues. All of it is important. Everything that I'm talking about in this set of lectures, you guys need to know, not just now, not just for the next test, but actually throughout the rest of the semester, because we're going to be talking about tissues all the way through until the very last day in class. So this material will be used quite frequently. So let's go ahead and get started. So you guys may remember from our introduction, tissues are groups of cells working together to perform a common function. Now, there are many different subtypes of tissues, but there are four basic tissue types. You want to remember the four basic types of tissue. We have epithelial tissue. Epithelial tissue serves to cover and line various surfaces or spaces inside the body. Good example are our skin. The top layer of our skin is an epithelium. We have connective tissue. This really sort of makes up the bulk of the body. This is going to be like packing material, adipose, tendons, that sort of stuff, bone, that kind of stuff. Muscle tissue, that should be fairly familiar, but we're going to find out there are actually three different subtypes of muscle. And then finally, we have nervous tissue. So this would include neurons and their supporting cells. So whenever I say ID basic tissue type, I'm talking about one of these four: epithelial, connective, muscle, or nervous. So just remember, when you hear that code word basic tissue type, you're talking about one of these four, only one of four possible answers. By ID specific tissue type, well, then there's like 20 to choose from. Okay, so you may remember last time we talked about cells. There are actually about 37 trillion cells in the average human body, right? That is a lot. They need to be organized and they need to work together, right? That's where tissues come in. So here's our official definition. A tissue is a group of similar cells and their extracellular products, right? Extracellular means outside the cell. So a group of cells, similar cells, and their extracellular products that perform a common function. They work together. So as I mentioned, our four basic tissue types: epithelial, connective, muscle, and nervous. So the tissues are going to differ from each other pretty dramatically, right? Bone and blood seem very, very different. And if you have a cup of blood sitting next to you and a bone sitting next to you, you would think these are two totally different things, but they're actually both considered the same basic tissue type. Bone and blood are both considered connective tissue. Okay, let's talk a little bit about this extracellular matrix, the stuff on the outside of cells. So remember, tissue, cells, plus extracellular matrix. Well, what is this stuff? Right, a substance produced by the cells, right? It is specific, so the extracellular matrix is different for each different type of tissue, and they can contain things like proteins, particularly protein fibers. They might include salts or electrolytes, water, other dissolved macromolecules, and they're always located on the outside of the cell. Okay, so there's a little table here that gives you the basic rundown of all the tissue types. So when you are done with this chapter, this is an excellent resource to come back and study this because it gives you lots of specific information. So what are the basic tissue types? Yes, definitely need to know that. What are their general characteristics? Yes, you do need to know that. What are their general functions? Yeah, you need to know that. Primary germ layer derivative. What this is basically describing is, where does this come from in embryological development? This you don't need to know because remember, in this class, we're focusing on adult anatomy, so you don't need to worry about this third column. So over here, example subtypes and their locations. I want you to have a general feeling for this right now, right? Would you need to know that you have simple columnar epithelium in the inner lining of the digestive tract? Yes, when we get to the digestive system. Right now, not so critical. What I would do for the basic tissue types and the specific tissue types we're going to talk about in this lecture, just learn one location, just so you have some idea where you might find this, because that will also kind of help you remember the function of the basic types and specific types of tissue. But when we cover an individual section, right? When we actually cover that section, that system in the body, yeah, you're going to need to know what all the different tissue types are, where exactly they're located, be able to identify them. So that is coming, but we're not there yet. Luckily, you don't have to worry about this until, oh wait, next time, because the next lecture is the integumentary system. So you will need to know all the specifics of the integumentary system, but for the next lecture. Okay, so the first of the four that we're going to talk about, one of the big groups is epithelial tissue. So epithelial tissue will line every surface of the body and every cavity, like so the insides of your intestines, the outside of our skin. This is all going to be epithelial tissue. Most of the glands, most of the exocrine glands of our body are derived from epithelial tissue. So this will include things like, like salivary glands, mucosal glands. They're going to be associated with epithelial tissue. One of the characteristics of epithelial tissue, it has relatively little extracellular matrix. So there's not a whole lot of stuff outside the cells, which means that this tissue is mostly cells, right? Because tissue is cells plus the extracellular matrix. Well, if you don't have much extracellular matrix, you're mostly cells. Okay, so characteristics of epithelial tissue, right? Cellularity, we just described this. It's composed mostly of cells, relatively little extracellular matrix. Now, in many other tissue types, it's that matrix which are binding the material together, keeping the tissue held together. Because we don't have a whole lot of that here, there's going to be a large number of intracellular junctions. So these are going to be structures that help bind the cells together, and we'll take a closer look at those in a little bit. Now, we have another characteristic of epithelial tissue called polarity. One surface is going to be different than the other. Like on our skin, this superficial surface, to use fancy anatomy terms, this is facing nothing, right? It's just the world, the end of me to the world. So the top or superficial portion of the epithelium is called the apical surface, sometimes called the free surface because it doesn't have any other part of you attached to it. Now, where this connects to the rest of my body is called the basal surface, or the bottom or the deep portion, right? So that would be the basal surface attaching to the underlying connective tissue. Okay, other characteristics, right? Attachment, the basal surface, right? The bottom of the epithelium will be attached to a thin basement membrane. So remember I said that there's relatively little extracellular matrix? Well, here in epithelial tissue, that basement membrane, that's it. That's all your extracellular matrix. And by the way, I'll have a picture of this that I'll go over in a second. Right now, I just want to understand the terms and the characteristics. Okay, so basal surface is attached to a basement membrane, which is an acellular structure produced by both epithelial tissue and the underlying connective tissue. So anyway, this basement membrane is partially made by the epithelium and is partially made by the connective tissue underneath. Again, I'll have a picture of that in a second. Now, another characteristic, avascularity. When you see the word A in front, it means not. So epithelial tissue is not vascular, which means it does not have blood vessels in it. So epithelial tissue lack blood vessels, so they're going to have to get their nutrients from deeper tissues that do have blood vessels. Next characteristic, epithelial tissue is innervated. Innervated means nerves are present, right? Nerves service this tissue. So if you touch an epithelium, you might feel it, right? It might have sensory receptors there, right? Which are connected to neurons. The neurons are going to be connected to your brain, so you can be consciously aware of somebody touching you. So epithelia are rich in innervation, or richly innervated, meaning they have lots of nerves to detect changes in the body or the environment. Now, can you think of an experience that actually sort of encapsulates these last two characteristics of epithelial tissue? Just think about that for a second. What am I talking about? Paper cut. Think about a paper cut, right? A paper cut is just a very superficial cut in your skin. Do paper cuts bleed? Not really, because they're only damaging epithelial tissue. They're not deep enough to go down into the connective tissue where you have blood vessels. Do paper cuts hurt? Yeah, they do hurt. Why? because epithelium, they may not have blood vessels in them, but they are highly innervated. So that's a good little phenomena to help you remember two of these characteristics of epithelial tissue. Now, another one, epithelial tissue tend to have a very high regenerative capacity, which means the cells can replace themselves. So they're going to be dividing rapidly. Good example of is our skin. You may know the top layer of our skin is dead, right? But the bottom layer of that epithelium is very much alive, and those cells are constantly dividing, multiplying, and then they go up and up and up and up, and eventually they die and they're sloughed off our body, right? So we need to be constantly replacing those cells so they have a very high regeneration capacity. This is good and bad. It's good because they're able to replicate and grow. So these tissues tend to do fairly well at healing themselves, right? You get skin damage and it usually heals in a few days, right? It's not a big deal. Anyone who's had tendon damage, right? You tear a ligament or a tendon, that takes forever to heal because you have different characteristics in that connective tissue. So epithelial tissue have a high regenerative rate. That's good because you can heal a little bit faster, but it may also prove to be a problem because if you have cells that are rapidly dividing, it means it's more likely that they could lose control of their cell cycle. What happens when cells lose control of their cell cycle and they start dividing uncontrollably? That's a tumor, right? If those cells then become invasive and they start colonizing other parts of the body, right, then it's cancer, right? So that's not good. So many cancers are derived from epithelial tissues. Okay, here is the picture that I promised. What we have here is an epithelium at the top of screen, and below that is deeper connective tissue. Notice that in the part that says connective tissue, we do have blood vessels, right? So that is vascular. Notice no blood vessels up here in the epithelium. So this is that apical or free surface, the part that's facing the outside world or the inside space of a particular organ. The basal surface is opposite to that, where it's going to attach to the basement membrane. Right? So this basement membrane is partially made by the epithelium and partially made by the connective tissue. I'll have another picture of that in a second. So apical surface, basal surface, basement membrane. The sides, these are called lateral surfaces of these cells. Now, one thing I want to point out is sometimes this gets complicated because this is epithelial tissue. This over here is connective tissue, but you see that these two little guys right there? That's actually an epithelium because the blood vessel has an internal space. It has a lumen, it has a void. All structures of the body are lined. So even blood vessels have epithelium inside of them. So if I said ID basic tissue type with the arrow pointed here, you would say epithelial. If I said ID basic tissue type here, you would say connective. However, if I pointed to this cell that's lining the tube of this blood vessel and I said ID basic tissue type, you would say epithelial. If I put my little arrow on a red blood cell, we'll get to this in a second, but I'm just trying to make a point here. If I had my pointer on a red blood cell and I said ID specific or basic tissue type, you would say connective because blood is connective tissue. So we got to keep kind of a close eye on what's going on here. There are lots of little detailed points that we don't want getting away from us. All right, this is a picture I stole from another source because it is a little bit more information. We can still see the apical surface, the basal surface, the lateral surfaces. We can see that the epithelial tissue is indeed innervated, which means our nerves are present, so we can get sensory information from this epithelium to our brain. You can see that down here, right, this is the connective tissue. It is vascular. We do have blood vessels down here. And now we see the basement membrane which is separating the epithelial cells from the connective tissue, actually has two components. It has the basal lamina here, and it has the reticular lamina. So basal means base or bottom. Reticular, you're going to see this word a lot in this class. Perhaps you heard of a reticulated giraffe or reticulated squirrel. So reticulated means woven or net-like. So if you've seen a giraffe, it's got that kind of crackle pattern on its side that looks kind of web-like. It looks a little bit like a net. So that's why they call it a reticulated giraffe, because it has that sort of web-like or the web-like appearance. Anyway, it's just sort of describing the protein's fibers in this area. But my point is, basement membrane is divided into two subcomponents. The basal lamina is created by the epithelial cells. The reticular lamina is actually created by the cells down in the connective tissue. So they work together to make the shared structure, the basement membrane. So this part here, the basal lamina, right, that is the extracellular matrix. Very small amount compared to everything else. Basal lamina is the extracellular matrix of epithelial tissue. Okay, moving on. So what does epithelial tissue do, right? Physical protection. Protection from dehydration, protection from abrasion, and protection from both chemical and biological agents. What are we talking about? Toxins, we're talking about bacteria, viruses, that kind of stuff, right? Keep that stuff outside of our body. We have a very thick epithelium on the outside of our body, which helps keep water in, helps keep dirt and pathogens out, right? We also have epithelium lining our digestive tract. Again, we want to keep pathogens out, but we want to be able to bring in various important nutrients in, which brings us to our second characteristic here, right? Epithelial tissue is selectively permeable. Why? Because you're going to have that apical surface and that basal surface, which is made of plasma membrane. And if you remember from our second lecture, the plasma membrane is semi-permeable. So epithelial tissue can control the flow of material from the apical surface down into the basal surface or vice versa. So we can control what enters and exits the body using epithelial tissue. Okay, secretion, right? Secretion. So many epithelial cells are part of glands, right? Exocrine is a particular type of gland. So examples would be sweat, oil, saliva, right? These are all made of cells in epithelial tissue. I mentioned that epithelial tissue is highly vascular, so it's important for sensation, right? Skin has a lot of touch receptors in it. Also, many of our most important senses, the actual structures that allow us to understand what's going on out in the world, are actually epithelial tissue. So the retina, the light sensitive cells in the back of our eye, that's part of epithelial tissue. Taste, right, our gustatory cells, these are part of an epithelium. The hair cells inside the cochlea or the inner ear is part of an epithelium. Sense of smell, and so on and so forth. Our sense of equilibrium, right? The cells that actually detect these things, light, sound, position, right? They are actually epithelial cells. Okay. Specialized structures. So the basement membrane, talked about this. This is gonna help organize and attach the cells of the epithelium to the structure. You're gonna find it between the epithelium and the connective tissue. Half of it, the basal lamina, is made by the epithelial cells. The reticular lamina is made by the underlying connective tissue. It will act as a barrier itself to help control the flow of materials across the membrane. So in most cases, material has to get through the apical surface, it has to get through the basal surface, then it has to get through the basement membrane in order to enter the body. So in certain circumstances, even the basement membrane also plays a role in regulating what is going to enter or exit the body. Okay, so I mentioned that epithelial tissue, you have cells all lined up together to make a layer. Now, in order to ensure that the cells just don't float away or separate from each other, there are special structures to bind cells together, to lock them together. And these structures are called intercellular junctions, right? Something that's going to connect one cell to another. And we're going to talk about 4 types of these junctions. We have tight junctions. They're called tight because they help exclude water, and we'll talk about why that is in a second. We have adhering junctions. To adhere to something is to cause them to stick together. So one of the main functions of adhering junctions is to physically bind one cell to another cell. Desmosomes and hemidesmosomes. A hemidesmosome is simply half a desmosome. Think of desmosome like a rivet. If you ever have worn jeans that have that little metal button looking thing on the side, that's a rivet. You find these at points of mechanical stress. Right? In your jeans, you have a little rivet there because you catch your pocket on a door handle or something like that and your pants tear open, right? You don't want that to happen, so that's a point of reinforcement. Gap junctions. So this is a junction that binds two cells together that actually has a hole in it. It has a gap, and actually material can pass from one cell to the other, and that's very useful for communication. Okay, so here is our epithelial tissue. Here's our connective tissue, the basement membrane. Now let's take a blow up right here, and we can see our epithelium. We have the apical surface, we have the basal surface, we have the lateral surfaces, and now we can see our various intercellular junctions. Now this is important. Tight junctions are closer to the apical surface because that's part of their function. Remember, the function of tight junctions is to prevent water from passing between these two cells, right? It's a tight junction, it's watertight. For instance, if water and water-soluble materials are, like, say, inside the lumen of your intestines, if that water could simply squeeze in between your cells, you would have no way of controlling what gets into or out of your body. It would be very difficult to control what's going on. So in order to ensure the material passes through the apical surface, passes through the cell, passes through the basal surface, ensuring that the epithelium is semi-permeable, we have to have a way to make sure they can't just go around the cell. And that's the job of the tight junction. Think of this as like a stitching or seams in your clothes. It's not super strong, but it very tightly binds these membranes together so water cannot squeeze through. That ensures that the epithelium is semi-permeable. Water just can't go around. Material just can't go around. Now, a little bit inferior to that, but beneath that, you have this guy right here, which looks a little bit like a belt, like a big, heavy leather belt going around the cell, right? This is the adhering junction, shown here, adhering junction. So the book describes this as a set of purse strings. I think it makes more sense having a big, heavy leather belt, because what this will do is this will provide structural support for each cell. Each cell gets a heavy, like, weight belt wrapped around the middle, and one cell will have its own adhering junction, and then another cell will have it, and then those adhering junctions are connected to each other. So that is an internal belt on one cell, internal belt on the other cell, and the two are bound together. So you can think of it, if you were to do this sort of weird thing, if you can imagine like three people standing next to each other, and they all had belts, and they would loop one belt through the belt of the person next to them, they would kind of all be stuck together. It's kind of a funny thing to imagine, but that's the basic functioning of the adhering junctions, to actually physically bind cells together so they don't become separated. Right? The tight junctions aren't going to be very good at doing that. They will block water, but they're not really going to keep the cells tightly bound together. Okay, our next one here are our desmosomes, here and here. Down here, we have a hemidesmosome. So desmosomes connect one cell to another cell, bolts them together just like a rivet or like a bolt. A hemidesmosome is going to connect a cell to the basement membrane, right? There isn't another cell on the other side, so the hemidesmosome is just connected to that basement membrane. So you will find these structures at points of stress. If you have lots of flexing in your skin or someplace where it's stretched a lot, there's lots of potential for damage going on, you're going to find desmosomes there just to keep the structure secure where it's encountering extra forces. Another thing to point out is right, you have these protein structures connecting both halves, the hemidesmosome, and then you have these filaments coming off, these intermediate filaments, if you remember those from our cytoskeleton. What this actually does is these intermediate filaments connected to the hemidesmosome will actually connect the cytoskeleton of one cell to the cytoskeleton of another cell. Now those two skeletons are locked together. That also provides structural support. So we have anchoring not just at the surface, not just at the plasma membrane, we also have anchoring between the cytoskeleton of one cell and the cytoskeleton of another cell, just another level of reinforcement. Our final intercellular junction down here is our gap junction. And in the gap junction, you're going to have these things called connexons, which are little tubes that pass from one cell to the other. And it's just an open passageway. So cytoplasmic cytosol can be exchanged in between the two cells. This can be very important for communicating in certain types of cells, cardiac cells, smooth muscle cells are going to communicate with each other using this structure, but we'll come back to that later in the semester. So what's the kind of the wrap up? What's the takeaway? Tight junctions prevent water from passing, prevent materials from passing, so they ensure that an epithelium is semi-permeable. The adhering junctions provide structural support to the cell and the epithelium. Desmosomes are, think of it as like rivets or bolts, which provide extra strength in areas where you have lots of flexing. They also connect the two cell cytoskeletons together. Gap junctions are important for communication or sharing the contents of one cell with that of another cell. Okay, so I've already pretty much given you the spiel, but I have everything written out here in words just to make sure nobody missed anything, and I didn't miss anything. So I'll just go over this real briefly and then we'll move on. So the tight junctions, right, are found near the apical surface. They prevent molecules from traveling in between the cells. They act as gatekeepers, right? Make sure that the epithelium is semi-permeable. Adhering junctions are deep to the tight junctions, right, made of microfilaments, and they're going to act as purse strings or belts that will protect the apical surface of the epithelial cell. Desmosomes, snaps, rivets, bolts, they appear at locations where you have lots of mechanical stress in the epithelium, right? They have a protein plaque that just means sort of a hard solid mass that's going to connect to the rest of the cytoskeleton. Right, particularly those intermediate filaments, which if you guys remember from the previous lecture, these act like steel cables and also provide structural support. Gap junctions have a fluid-filled channel that allows for materials to pass in between cells. Very important for communication, flow of ions, chemical signals, things like that. Okay, now we're going to talk about classification of different types of epithelial tissue. Many different types of epithelial tissue, and we have a couple of different ways of classifying them. So the first two that we're going to talk about are the number of cells in the epithelium. Do you have one layer of cells? Do you have many layers of cells? And what are the shapes of those cells? Are those cells flat like pancakes? Are they kind of cube shaped? Are they square? Or are they tall columns? So by classifying cells based on the number of layers, we really have two and like a third extra kind of funny one. So let's take care of the easy one, or the clear one first. A simple epithelium means you only have a single layer of cells on top of the basement membrane, a single layer of cells. All of them have an apical surface. All of them have a basal surface. Nice and easy. All right, a stratified meaning layers, a stratified epithelium has at least two or more layers of cells. So not all the cells will reach the apical surface, and not all the cells will reach the basal surface. Only the bottom layer will touch the basement membrane. Only probably the top layer is going to touch the apical surface, right? You clearly have one stack, another stack, another stack. All right. Easy enough to understand. Now, this third one is a weird one. So pseudo, the word pseudo means false. So a pseudostratified epithelium is really a simple epithelium, but it looks like a stratified epithelium. So it's a falsely stratified epithelium. So to put this more simply, a pseudostratified epithelium, you only have one layer of cells, but it looks like two or more. And I'll give you tricks for figuring this out. So in a pseudostratified epithelium, all the cells will connect to the basement membrane, but not all of them will reach the apical surface. Okay, so let's take a look at the easy ones first. Simple epithelium, we have a basement membrane, one layer of cells, we're done. Down here, we have a stratified epithelium. Here's our basement membrane. All of these cells of the basal layer do touch the basement membrane, but they do not touch the surface. Then we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, or 16 layers up, we have a layer of cells that actually touch the apical surface. They're actually exposed to the outside environment, right? So those cells obviously do not touch the basement membrane. Okay. We'll talk about pseudostratified in a little bit. Now let's talk about epithelial based on the shape of the cell. So we have squamous. Squamous is just a fancy word for flattened. A squamous cell is squashed flat like a pancake. Sometimes they have little irregular shapes. Cuboid, it's cubic in shape. They're about as tall as they are wide as they are deep. So more or less equal on all sides. Columnar means it's like a column, right? Columns are thin and tall. So a columnar cell is taller than it is wide. So squamous cell, cuboidal cell, and columnar cell. Now, is there a gray area between cuboidal cells and columnar cells? Are you going to get some columnar cells which are kind of short and they're about as tall as they are wide, and a couple of cuboidal cells which may be stretched out a little bit? Is there a gray area in between? Yeah, unfortunately. However, there is a way to tell the difference, and the key is to look at the nucleus. So even this picture shows that a little bit. In a cuboidal cell, you tend to have a large, centrally located nucleus. So right here, you have a circular, centrally located nucleus. Often big, not always, but frequently it's big. So cuboidal cells, circular, centrally located nucleus. Columnar cells tend to have an oval, like an egg-shaped, an oval nucleus, which tends to be closer to the basement membrane. Not always, but usually. So if you can't tell, is that a cube or is that a column? Look at the nucleus. Centrally located and round, that's cuboidal. If it's oval and near the basement membrane, that's columnar. That's how you tell the difference in those gray areas. So here we have a nice breakdown of the different types of epithelium. Each of the epithelium is going to be characterized by both the shape of the cell and the number of layers. So simple epithelium, this is a group that only contains one layer. Simple squamous, simple cuboidal, simple columnar, non-ciliated. So this would be one layer of columnar cells that do not have cilia. And then you have simple columnar ciliated, one layer of columnar cells that all have the wavy hair things. Then we have our different types of stratified squamous, keratinized. We'll talk about that in a second, but basically keratin is a tough fibrous protein that's useful for protection and to seal in water. Stratified squamous, many layers of flat cells that do not have keratin. Stratified cuboidal, stratified columnar. Right. Other types, pseudostratified, we'll talk about that in a little bit. Okay. Actually, we'll talk about it now because here is the picture I stole from another source because it does a good job explaining what's going on. So we have a simple epithelium, one layer of cells. We have a stratified epithelium, many layers of cells. And here's that funny pseudostratified that I talked about. When you look at this, or when you look at a histological slide in the microscope, remember you generally can't see the lateral surfaces. You're not going to see the plasma membranes very clearly. you look mostly at the nuclei. It looks like that this nucleus has got this guy on top of it, this one's underneath, this one's on top, this one's on bottom, this one's way up high. So it looks like there are multiple layers of nuclei, which makes you think there are multiple layers of cells, but it's not true. Each of these cells touches the basement membrane. However, not all of them reach the apical surface. Like this guy here, he peters out halfway through, right? He doesn't reach all the way up. This one goes from the basement membrane all the way to the apical surface. But that guy, and that guy, and that guy, and this guy do not. So just keep an eye out for that. It looks like multiple layers of cells, but there's actually only one. Pseudostratified. Excuse me. Down here, classification of epithelia based on the shape. Squamous, meaning pancake, cuboidal, meaning cubes. Notice centrally located, circular nucleus. Columnar, you're going to have oval nuclei closer to the basement membrane. Okay, so I am going to show you some pictures of each of these types. These slides include information where you will find this. This is more kind of for your information at this point because it will help relate structure and function, help clear in your mind. I'm not going to specifically ask you on where you find these things until we get to that specific body system. Okay, so first up, simple squamous epithelium. So one place you will find this is on the amnion. So this word may be a little familiar to you, amnion, like an amniotic sac, right? When we were in utero, we had a simple squamous epithelium covering us, right? Inside of that was the amniotic fluid. Outside of that was the uterus, right, of the mother. Another common place you find simple squamous epithelium would be in the inner lining of blood vessels. So remember, blood vessel is a tube. You got an empty space in there. That needs an epithelium to line it. So you will find an epithelium, a simple squamous epithelium lining blood vessels. It actually has a specialized name called endothelium, but we'll talk about that once we get to blood vessels. All right, next up, simple cuboidal, right? One layer of cubic cells. So here we go, right? That is the apical surface. That is the basal surface. We have a large circular centrally located nucleus, mostly circular, right? That one's a little bit oval. This one, these two are slightly oval. It's the real world, so not everything is picture perfect textbook. One layer of cuboidal cells, large centrally located nucleus. Now, this happens to be in the kidney or in the nephron. This is where the body will separate, filtrate from blood, which will eventually become urine. Now, one thing I wanted to point out, just to kind of remind you guys about the 2D versus 3D histology thing, this picture here is of a tube. It doesn't look like a tube because you would expect a tube to look like that, right? You'd expect to see circles, right? But remember, this is a three-dimensional object. If this is your tube, if this is your nephron, and you cut it like this and look at it edge on, you would expect to see a circle. However, this image is what's called a longitudinal section, which means the tube has been cut in this direction and then opened up, so it looks like a channel. So just don't forget about those kind of funny orientation things. Okay, next up, non-ciliated simple columnar epithelium. So one layer of columnar cells, no cilia present. One common place you find this is going to be in the intestinal tract. Right? What's the job of the intestines? Finish chemical digestion of our food and absorption into our body. So it kind of makes sense you would want one single layer of cells, right? Because a lot of material has got to be absorbed from the lumen of your intestines into your body. So a simple epithelium makes some amount of sense here. These little white pockets, these are structures called goblet cells because they look a little bit like a wine goblet and they're white in color and they actually produce mucus. Also, here is that brush border we talked about. Notice that you cannot see individual hairs. That's how you know those structures are microvilli and not cilia. If you saw individual hair-like structures, you would know it's cilia, right? You cannot see individual hairs, so that's how, when you look at this, you can look one layer of columnar cells, simple columnar epithelium, non-ciliated. Here, we have one layer of columnar cells, and we do see individual hair-like structures. So this is simple columnar epithelium ciliated, or ciliated simple columnar epithelium. And an example of a kind of place where you would find this would be in the uterine tubes, the fallopian tubes is kind of the old term for these, right? Why do we need cilia here? Well, it's the job of these cells to help waft the egg from where it was released from the ovary, right? This is the ovary, it needs to get wafted down to the uterus for implantation. That's the job of those cells. Now, non-keratinized stratified squamous epithelium. So as I mentioned, keratin is a tough fibrous protein. It provides lots of structural strength, structural support, and it is waterproof. So that's one of the important jobs that keratin does in our skin, is to lock water in so we don't get dehydrated. But this is non-keratinized, which means there's no keratin present, which means it can't lock water in. So we're only going to find this epithelium in moist places, or places that have their own source of moisture and lubrication. Stratified squamous means you have many layers of flat cells. So here we have the basement membrane, right, our basal layer of cells, and we have many layers of flat cells all stacked up on top of each other until we get to the top surface. So many layers of flat cells. Non-keratinized means that the cells are alive, because when a cell fills up with keratin, it dies. We'll talk about that in a second. And it needs to be kept moist. So the kinds of places you find non-keratinized stratified squamous epithelium will be the inside of the mouth, inside the vagina, in the anal canal, places where you have moisture. You need those layers of cells because you need protection from abrasion, right? When you eat, you're sticking stuff in your mouth and you're grinding it up, right? If you only had one layer of cells there, that really wouldn't give you very much protection. But having lots of layers of these cells does protect you very well. Same story at the other end, right? Having a bowel movement, you need some protection there because you have abrasion, you have mechanical action going on there, and there's a huge amount of bacteria in fecal matter, so you need to protect yourself from it, right? Vagina is the birth canal, right? A baby might come out of that. You're going to need a fair amount of protection from that occurring. Okay, now we have our keratinized stratified squamous epithelium. So these cells near the apical surface are filled with this protein, keratin, this tough fibrous protein. And when they fill up with this protein, the cells actually die. So they don't actually have a nucleus, they're dead. So that's why that top layer of your skin, you're always shedding dead cells. This is what we're talking about here. Keratin is watertight, so your skin is dry, right? The inside of your mouth needs to be kept moist, otherwise it's really uncomfortable, but your skin can be just fine when it's dry, and that's because of the keratin in these outer layer of cells. So keratinized cells, many layers. These cells up here are all dead. The cells right here are in the process of dying, and these cells are in fact alive. And a separation between the pink and the blue, that represents the basement membrane. So this is actually a histological slide taken from what's skin from the palm of the hand or from the sole of the foot. We'll talk about that once we get into integumentary. But just so you realize, we have many layers of cells. That's the basement membrane. That's the apical surface. These cells are all alive. Once they fill up with keratin, they die, and they form that outer surface of keratinized stratified squamous epithelium. Do you need to remember those terms in that order? Yeah. I'm going to want keratinized, stratified, squamous epithelium, right? On a practical, I'm going to want keratinized, stratified, squamous epithelium. Don't say KSSP and abbreviate. No, I want the whole thing written out. Don't leave off epithelium. I mean, I might let you abbreviate epithelium if EPI, but don't just leave it off, because are you talking about connective tissue? Are you talking about epithelial tissue? Are you talking about nervous tissue? Are you talking about muscle tissue? Right? We need to be exact and explicit and complete in our answers. Stratified cuboidal epithelium. So here we're going to have 2 or more layers of cuboidal cells. So this is fairly rare in the body. Kind of places you will find this would be in the ducts of certain glands. Like when you look at sweat ducts leading out of sweat glands in the body, you will find stratified cuboidal epithelium. Now, stratified columnar, multiple layers of columnar cells, this is very rare in the body. Stratified columnar, very rare in the body. There's really only one place that we're even going to talk about in the body where you find this, and that's actually in one small section of the male urethra. So you're probably not going to encounter this unless we're talking about the reproductive system, and then only about the male reproductive system. So just remember, truly stratified columnar epithelium is quite rare. I'll just make a note of it now. Notice, no cilia. Now let's go to the next one. Pseudostratified columnar epithelium. This looks like multiple layers of columnar cells, but it is not. It's actually one layer of cells that have this kind of jagged appearance. Now, how do you tell pseudostratified from, well, if you think you see multiple layers of nuclei, you should ask yourself, does it have cilia? If it has cilia, remember, cilia means pseudostratified. Stratified columnar, right, not really a thing. Cilia are not really a thing on stratified columnar. Pseudostratified will have cilia. Is that always 100% true? No, it's really about 95% true, so I'm lying to you a little bit. But what's 95%? That's an A, right? And that's good enough for this class. So let's just leave it there. Okay. Ciliated pseudostratified, looks like multiple layers of columnar cells. You can't really tell the difference. If you see cilia, then it's pseudostratified. No cilia, just go with simple columnar. Now, another kind of dead giveaway about telling the difference between truly stratified and pseudostratified columnar, goblet cells. If you see those goblet cells, that's like an extra little kicker to say, yep, that is indeed pseudostratified. Remember, ciliated simple columnar is a thing, right? This one you found in the body, not all that rare. But remember, this is one layer. My little trick with the cilia is because if it looks like you have more than one layer. So remember those little details so you don't get caught. Now, the last one we have here is called transitional epithelium, and transitional epithelium is found in urinary bladder and in the ureters. And what kind of characterizes transitional epithelium is that the basement membrane and the cells, or I should say, the cells of the apical surface, the cells at the top, have many different shapes. All the other epithelium that we looked like, all the cells of the top surface all had the same shape. They were all cubes, they were all columns, they were all pancakes. Here, right, they're going to have lots of different shapes. You're going to find some that are squamous and some that are cuboidal and some that are going to look kind of columnar, and they're going to be, you know, cubes and triangles. You're going to see lots of different shapes. And the reason for this is because this epithelium is stretchy, because the way the bladder works, it isn't like a metal can that has a rigid structure and then it just sort of fills up with urine and then drains out and then you have air in there. It's not how it works. The bladder works like a balloon. So when the balloon is empty, it's all shriveled down, and then when urine enters it, it expands like a balloon. There's no air gap in there. So that space inside the bladder has to expand, which means the lining that covers that space also has to expand. So when you have a transitional epithelium that is relaxed, there's no tension on it, most of the cells are going to look kind of cuboidal or columnar. If you were to take that transitional epithelium and stretch it tight, like when the bladder is all full, and then you take a slice, all those cells will look squamous because they've been all stretched out. So how do you recognize transitional epithelium? cells of the apical surface are going to have kind of a jumbled appearance. Also, if you're looking through the microscope and you see a part where the tissue is stretched like this, most of the cells in that stretched area will look squamous. If you have it folded in like that, most of the cells around here are going to look cuboidal or columnar. Also, sometimes you will find binucleate cells, or cells that have two nuclei. The example is right here in the cartoon version, right? One cell, two nuclei. Also probably right here, one cell, two nuclei. You don't always see them, but it's just like another little indicator to let you know that you got it right. Whew. That takes us through all the basic types of epithelial tissue. Now we need to talk a little bit about glands, which are derived from epithelial tissue. So glands might be an individual cell or it might be a whole big organ, right? Their job is secretion. They're going to make some material and they're going to release it into the body. They can do things like produce mucin. Mucin is a glycoprotein. When mixed with water, becomes mucus. So the cell makes mucin, mixes with water, becomes mucus. Glands release things like hormones, antidiuretic hormone, testosterone, all that kind of stuff. Glands can produce enzymes. Salivary glands produce salivary amylase and help break down starch. And they can all produce other types of waste products. Two basic classifications of glands. We have endocrine glands, and we have exocrine glands. So the thing to remember is an exocrine gland, exo means on the outside. These glands have ducts, they have tubes. So when an exocrine gland makes something, it dumps it into this tube and the tube leads somewhere. So sweat glands in the skin. The gland produces the sweat gets dumped into a tube, a sweat pore, which then extends to the surface of the skin, and then it spreads out and evaporates. So a sweat gland is an exocrine gland. Exocrine glands have ducts, they have tubes. Endocrine glands do not have ducts, and what they do is when they produce a compound, they don't dump it into a tube, they dump it into your blood. So the bloodstream is the mechanism of transportation for endocrine glands. So classic example of that is hormone. Like your hypothalamus and your pituitary are endocrine glands because they're going to produce hormones, dump it into your blood, and the blood carries the hormone wherever it needs to go. So here's a basic structure of an exocrine gland. So when you see that exo, think duct, and there it is. So this is a set of tubes draining this exocrine gland. The outer structure is called, the capsule is made of a material called stroma. So bits of that capsule dive down deep and subdivide the gland. Those subdividing structures made of stroma are called septum. The word septum literally means a divider, a thing that divides. So this specifically would be capsule. That part right there specifically would be septum, but they're both considered stroma. It's usually made of like a fibrous material. Provides just the structural foundation for the gland. Now, this little area here, which contains the epithelial tissue, which is making the substance, this whole structural unit, all these little red berry-looking things put together is called parenchyma, just a set of associated structures. If we take a zoom-in look at one of these little red berry things, we get this guy here. We have the duct, because it's an exocrine gland, and we have a structure here called the acinus. Acinus or an ascus is just a fancy word for a bag or a sack, because it looks like a little purse. So here are the cells. Here are the exocrine cells. Notice that this is an epithelium because they're lining the space. These cells are going to produce the material that gets dumped into the duct and drain out of the organ to wherever it needs to go. This was a salivary gland. The duct is going to open up into the oral cavity. Okay, so we have the axis, the acinus, which is the actual secretory cells. They're going to dump their material into the duct. Excuse me. Other large structures, and that's drained by one of these main ducts is called a lobe. So this structure here is a lobe. Lobes are subdivided into smaller structures called lobules. So this is also a lobe right here, right? It can be broken down into one lobule, another lobule, and a third lobule. So we have the whole gland itself covered on the outside by a spotted stroma. One specific area of the gland is called a lobe. Lobes get subdivided into lobules. One lobule, another lobule, third lobule, fourth lobule. Right? If you look at one unit of secretory structures, that's a parenchyma. You look at one of these little berries, we have an acinus drained by a particular duct. So we will see this structure, this generalized structure, many times as we look at different types of exocrine glands throughout the semester. Now, these glands can take on many different shapes. So I have, what is it here, like 8 different ones. I'm not going to make you memorize each of these, but it is important to understand the kind of shapes that they take, because when you look at a histological slide, you're going to need to know that this and that, which look totally different, are still all both exocrine glands. So just to go through this briefly, but I won't test you on how do you tell one from the other. Simple tubular, just a straight tube, right? Branched tubular, just straight branches. Simple coiled, we're going to see a lot of those in the skin. So they're going to be basically, think of it as a big long piece of string crumpled up into a ball. It's kind of a big mess. Simple acinar, this means it has that sac or that bag at the end. It's not just a straight column. Branched acinar. Compound tubular, compound acinar. That means you're going to have branches off your branches, right? Compound tubular acinar, you're going to have a mix of both. So I'm not going to make you tell the difference between a compound tubular and a compound tubuloacinar, but I just want you to know those possibilities are out there because it helps you identify what you're looking at when you look at the slides. Whew. All right, that covers 1/4 of this lecture. Remember, I told you this was a long one. In reality, we're more like 1/3 of the way through. Ooh. Hopefully you guys didn't hear that. There's a fire truck going by. Epithelial tissue and connective tissue are really, really big sections. However, nervous and muscle go very, very quickly, so it's not nearly so bad. But yeah, this is a bit of a doozy, and I will see you guys when we do part 2. So take care.